Greetings, everyone. Welcome to today's webinar, Pharmaceutical Industry. It is my pleasure to be together with you all again today. Just some housekeeping notes to share with you. All attendees are in listen-only mode, and this webinar is being recorded. I am Lori Sjogren, Community Program and Services Director for the National Ataxia Foundation, and I'll be assisting with today's webinar along with our Communications Manager, Stephanie Lucas. Questions for our webinar speakers can be typed in the questions tab found in your control panel, and we will answer as many questions regarding this topic as time allows during our time together today. So as your questions arise, please send those to us. So before we begin today's webinar, our Executive Director, Andrew Rosen, would like to say a few words. Welcome, Andrew.
Thanks, Lori. Hi, everyone. Lori and I are coming to you from Minneapolis. I just very quickly, I just wanted to say a couple of thank yous here. So this webinar today is going to wrap up a series of webinars, probably 15 of them, that we wish we had been able to present live to you all in Denver this past March. And as you know, we had to cancel the conferences due to COVID-19. But I just want to say thank you to, first off, everyone who's on the line today. There's almost 220 of you now, and we know that many more watch the taped version of these, for hanging in there with us for the last 3 months and for tuning into what we think is some really good content and really important messages that we've been delivering. And so thank you for joining us for these. To our panelists, of course, Dr. Wilmot and Mike Curtis and Melissa Beiner, all friends of NAF, and we thank you for your time today. And then lastly, just a special shout out to Lori Sjogren. Lori has, again, has done like 15 of these in the last few months. And these are not a simple little process to put together. Not only using the technology, but making sure all the panelists are ready to go and slideshows are ready, etc. This is a big deal. And Lori, thank you from all of us at NAF for all the work you've done. So with that, enjoy. I'll turn it back to you, Lori.
Well, thanks so much, Andrew, for those kind words. So as Andrew mentioned, for today's webinar, we have two representatives from Biohaven Pharmaceuticals and Cadent Therapeutics. Both companies have programs developing drug therapies for ataxia. On today's panel is Melissa Beiner from Biohaven Pharmaceuticals and Dr. Michael Curtis from Cadent Therapeutics. And Chip Wilmot from Emory University will be facilitating today's discussion. Now I'll turn it over to Dr. Wilmot. Welcome.
Thanks very much for hosting this wonderful webinar. We were going to do this, as Andrew said, as part of the annual meeting. And I really look forward to helping to facilitate a kind of roundtable where we find out a little bit about things from a pharmaceutical industry perspective. I am not going to be talking today. I'm just going to be facilitating. All of you out there probably happy to hear that. But we do want to encourage questions from you. This is an opportunity to talk with two representatives of companies that are extremely involved in bringing therapeutics to ataxia. This is an extremely wonderful time in ataxia research where we've gone from not being able to even measure ataxia 10 years ago or more, around in that time, to developing enough knowledge about ways of measuring ataxia, which we're still working on, and knowing enough about the diseases where we can actually do clinical trials. So it's a very exciting time. The clinical trials are here, and we're so excited about that, and excited today to hear from Melissa and Mike. So I'm going to go ahead and turn it over to Mike, who will talk to us about his stuff going on at Cadent Therapeutics. Go ahead, Mike.
Great, thanks, Chip. Thanks, Lori, and a special thanks to Sue Hagen. Sue was one of the first people we met in the National Ataxia Foundation about 6-7 years ago, when we first had this idea of trying to regularize neuronal firing as a way to improve movement coordination in patients suffering from ataxia. So just a little bit on Cadent Therapeutics, and if the slide goes forward, beautiful, that's a good start. So we're developing therapeutics for several different indications. One side of our company works on mood disorders and cognitive disorders, specifically in the context of schizophrenia. Won't talk about that today. And the other side of the company works on treatments for movement disorders. And we'll talk a little bit about the history of how we got into movement disorders and the approach that we're using. We're supported by a group of investors who've been with us since the beginning, almost 7 years now. And we also have put together a really incredible team of drug developers to help push our therapies from discovery, right, all the way through the clinic. So first, a little bit of biology and a little bit of medicine of why what the approach is that Cadent is taking and why we think it's going to be important for the treatment of ataxia. So what we're looking at here is a piece of cerebellum and on the top panel, and on the bottom is a zoom in on a special set of neurons known as Purkinje cells. Now, these neurons are really important for keeping the rhythm of the brain. So in everyone, they fire your whole life at a very high rate, and your brain uses that sort of as a timekeeper to keep everything in sync. And when people think about Purkinje neurons in cerebellum, they have historically thought about it as controlling movement and allowing smooth movement. I think what Jeremy Schmahmann at MGH would actually argue is that it actually smooths everything, right? It smooths mood, it smooths all facets of brain function. We can measure, and this is some work that we've done in rodents, we can measure the firing frequency or how often the neuron is firing using what we call electrophysiology. And if we measure a healthy neuron, a healthy Purkinje neuron, it fires at a very regular rate. Again, very consistent your whole life. If we then introduce ataxia, and this is into a rodent, into a rat, if we introduce ataxia into a rat, what we see in Purkinje neurons is misfiring. So we see what we call asynchronous firing. So the neurons are not firing correctly. And we think that this asynchronous firing or incorrect firing in the Purkinje neurons leads to balance and stability disability that we see in patients with ataxia. So, well, how can we correct this? Well, there's several approaches that are being pursued. The approach that we're using is to use the ion channels. So ion channels are in all neurons, and they control the flow of the electrical impulses. So, right, your brain is really just a series of electrical connections, and the ion channels control and regulate those electrical connections. What we're developing is a modulator of one of those channels. It's called the SK channel, and the SK channel plays an important role in maintaining the regular firing of Purkinje neurons. So if we think about the cerebellum of someone with ataxia and their Purkinje neurons that are having trouble firing consistently, if we can upregulate or turn up, essentially, if you think about it as a rheostat or a dimmer switch, if we can turn up the current in the SK channel, the hope is that we can regularize Purkinje neuron firing. Now, we won't go through all the data that we've generated preclinically, but we can show that our compounds can actually increase current through the SK channel. And in rodent models, we can actually improve motor control and coordination. So what we've been working towards for really the past 6-7 years is establishing that fundamental science, and now we're ready to move that program into the clinic. And when I was talking to Sue about being part of this panel, and the fact that we've known each other for going on 7 years, part of her wonder is like, what the heck have you guys been doing, right? We first talked in 2013, and now it's 2020. So just to give you a little bit of perspective on what it takes, right, to bring a compound from discovery all the way to where we are now, which is at the doorstep of a phase 2 clinical trial in ataxia. The compound that we're talking about at the end of this pipe here, 2019, is CAD1883. And that started way back in 1989 with a company called Neurosearch. And this company was created to develop treatments for Huntington's disease, Parkinson's disease, other movement disorders. Around 2005, they actually screened this SK channel. The whole idea of Purkinje neuron regularization and using that as a mechanism to improve patients with ataxia started right around then. They did a screen, and some other webinar we can talk about what screening is all about, but they discovered the first compound that could actually regularize these channels or affect these channels. They then went on from 2006 to 10 to make 600 compounds, right? So they're optimizing, changing chemistries, trying to figure things out. And what we're trying to figure out is, one, we want to be able to give a safe molecule, and we want to give it as a pill, so orally, right? We want it to stay in the circulation of a patient for at least 24 hours. One of the challenges with compounds that we use in neuroscience is they have to get into the brain. And the brain is really designed to keep things out, right? So we need to find drugs that can actually get into the brain, and then ultimately that could control disease. So the first potential drug was discovered in 2012. That didn't have the brain penetration that we needed. we bought the compound from a company called Saniona. Saniona came out of NeuroSearch, the long story there, but that's where we first met Sue in National Ataxia Foundation when we were Ataxion. We then went ahead, made about 600 compounds. That led us to our first compound that we thought we could take into the clinic. That's 1289. That we actually talked to NAF about that years ago. There were some issues with the potency, stability. We needed a different compound. We then went on to 1551. That was our third potential. That also ran into some troubles. We made another 200 compounds. So by now, we've made 1,400 different drugs to address all of the different parameters that need to be addressed to be a successful drug in the clinic. And that led us to 1883. In 2018, we took that into phase 1. Phase 1 is a healthy volunteer study. We were just really primarily concerned about safety. Can we safely give this drug? And the good news is we can. And then in 2019, we ran a proof of concept trial in another movement disorder called essential tremor and showed that we could actually reduce or improve disability in patients with essential tremor. And so here we are, 2020, ready to launch what we call Synchrony 1. So Synchrony 1 is a phase 2 proof of concept trial in patients with spinal cerebellar ataxia. A couple of the highlights, and working with the National Ataxia Foundation led us to much better understand the need in the space, what do patients suffer from, and then also the need for inclusion, right? And so we've designed this first POC study to really include pretty much all of the dominant spinal cerebellar ataxias. You can see through SCA17, and we had a long debate with Jeremy about the inclusion of ARCA1, which technically is not considered SCA, but a lot of the biology is very similar to what we see in patients with SCA. One of the things we'll talk about today is endpoints. In this trial, right, we're focused on SARA, and I believe many of you folks have been involved with actually participating in a SARA score. And one of the challenges we have is we need to show that we can affect the disease in a relatively short amount of time. You can see this is a 12-week study. I know Melissa will talk about some longer-term studies that the team at Biohaven are doing. But we need to see something in the first 12 weeks to show us that we're engaging the biology, so we're improving Purkinje neuron function, with potentially leading to a benefit to patients, right? Just improving Purkinje neuron function without helping improve balance, coordination, and control isn't that great, right? So one of the things that we've been doing on the side as we've been cooking along 1883 is try to figure out are there novel endpoints that we can use in addition to SARA to look at improvements in patients' ability to maintain balance and maintain motor control. And so we've actually worked with a company called Biosensics in collaboration with the team at MGH to do what we call an instrumented study. And we did it, and this is really hot off the press data. This is the first time we've shown it to anyone, but we're really excited about with the potential here. And what we've done, there were really two goals of this study. One is to correlate a quantitative measure. And so what the Biosensics system is a system of gyroscopes and accelerometers that can actually quantitatively measure balance and motor control, and then correlate that with the SARA score, which I think most of you have done. And what we've shown is that the instrumented measures, the quantitative measures, correlate really well with SARA. And what this means is that we might be able to eliminate the need for the neurologist to score a SARA. Now that's a little bit down the road, but even talk to anyone, Jeremy, Sue Perlman, I think Chip would agree, right? Getting 3 neurologists to agree on what a SARA score is challenging, right? It's a big part of getting a clinical trial off the ground is everyone to agree on the scoring. If we could remove that noise or that variation, I think we can improve the outcomes of these studies. What we're really excited about is also the addition of new endpoints. So in the middle panel, you can see we're looking at 2 measures of movement control. One is turn duration, right? So we know patients with ataxia have a little more difficulty turning around. Well, we can all see it, but what these sensors allow us to do is measure the rate of your ability to turn. And then what we could look for in an endpoint is improvement in turn duration. The other one is body sway, right? If we ask a patient to stand still, it's difficult to do. And we can measure the velocity of sway. We could imagine we can also measure the velocity of sway following 4, 8, and 12 weeks of therapy. If we see an improvement in sway, that's not necessarily, now we have a drug, but it sends us on the path to say we're doing something to the biology, we're improving the control, motor control, and in a longer term study will we actually have a clinically meaningful benefit. So these novel endpoints for us are really important because I think we'll talk about investors and how we raise money to run these studies. It's very important for us to be able to show early in clinical development that our drug is doing what we think it's going to do long term. And these new endpoints may give us a way to do that. So with that, I will hold off any questions and turn over to Melissa to provide a background on Biohaven.
Okay, so hello everyone. I'm Melissa Beiner and I'm the Director of Research and Development here at Biohaven. And I would love to thank the members of NAF for having me on this panel. And, I think Mike and I can both agree that working in this space with the National Ataxia Foundation, our key opinion leaders across the country who are the leading experts in the ataxia field, as well as the patient population, I think it, especially for people in pharma, it's an incredibly unique environment to work in because NAF is so collaborative with us, the neurologists across the country that are participating in our studies, our trials, are so collaborative with each other, and the patient community is really, really invested. So thank you to NAF and also to our KOLs, one of whom is our leader on this discussion, Dr. Chip Wilmot, for including industry in this, because I think the collaborative efforts of all of us are what are now leading to the huge advancements we're seeing now in the field of ataxia that have really evolved exponentially just in the last 2 years. So I'm going to go through these slides very quickly and very briefly so we can get to the panel. A lot of the people on the line probably know quite a bit about Biohaven because we're now deep into our second clinical trial in spinocerebellar ataxia. Biohaven is predominantly a clinical stage pharmaceutical company, meaning that we do clinical trials in humans at the phase 1, 2, and 3 level. We have a pretty broad pipeline. We have 6 products across 3 broad platforms. And of course, for today, I'm really going to hone in just on our platform for neurodegenerative and neuropsychiatric diseases. We are based out of Biohaven because there's a very strong academic background from the leaders of our company, 2 of whom trained extensively at Yale, which is how I came into contact with this company. I also did all of my medical training for my MD and also my residency at Yale. And then, the leadership has probably between them an average of about 20 years of pharma experience. We now actually have broadened our company to a much larger company to include an entire operations department, which can commercialize drugs at this point because part of our pipeline, a migraine pipeline, is currently commercializing our big drug there. So as I said, I'm going to hone in on our glutamate platform, which is a platform that we have to treat both neurologic and neuropsychiatric conditions. So the drug that we're using in our ataxia clinical trial for spinocerebellar ataxia is troriluzole, and it's a glutamate modulator. So what is glutamate? So glutamate is the main excitatory neurotransmitter or neurochemical in the brain. It's present in about 90% of brain synapses. So it's critical to normal brain homeostasis and normal brain function. What happens when there's too much glutamate in the synapse or in the brain in general is you get excitotoxic states, and that has been implicated in a whole host of disorders. And you can see on the left anything from ALS, where riluzole, a glutamate modulating agent, is the only FDA-approved treatment for that disorder, to spinocerebellar ataxia, which is where we're using our drug here. And the drug acts predominantly by not decreasing levels, but by normalizing glutamate levels in the brain to maintain that critical homeostasis. So this is just sort of a complicated cartoon to illustrate sort of how our drug works. Again, the drug acts by normalizing glutamate levels, but predominantly what it does is it increases glutamate uptake from the synapse. So the synapse is kind of that space where neurons or cells of the brain communicate. And the cerebellar ataxias, we're honing in really on the Purkinje cells and glutamate excitotoxicity in the cerebellum. And as we all know, there are no FDA-approved treatments for spinocerebellar ataxia. So as part of what we're going to talk about today is where do we stand in sort of the SCA clinical trial space and what guides clinical trial development in a rare disease space. So how do we align on trial design and how does this all apply to SCA? So this slide pretty much highlights the 4 major areas that are pretty important in guiding clinical trial design. And where this is important in SCA is that we don't have a lot of what's in that upper right corner, which is clinical trial experience. So There were a couple of studies out of Europe using the drug riluzole, again, a glutamate modulating agent that's been used in ALS to treat spinocerebellar ataxias. In the United States, there was virtually no clinical trial experience. We felt the evidence to use a glutamate modulating agent in spinocerebellar ataxia was compelling enough that we did launch and carry out the first registrational trial in SCA in the United States, which was our 201 clinical trial. And what guides that, the design of any of these trials, are outcome measures, which, again, to find sensitive and specific outcome measures, which Mike alluded to earlier, really you need some clinical trial experience in order to develop those. And those become very big facilitators of drug development down the pipeline, because other companies are going to be more interested in getting into this space the more we have an easier trial design. Natural history data is really critical in letting us know how long we need to keep these trials running, what's the duration of how long we need to look at these patients, because it guides us via the natural progression of the diseases. And then another important thing, especially that guides clinical trials, is having sort of biomarkers or quantitative assessments. And Mike talked about a little bit about the sensor studies, which are being developed to provide us with some quantitative guides to follow how patients evolve clinically in a trial through, all of these measures. And again, this is a really evolving field. So what do we know about natural history study? And this has come into play really very much with Biohaven, because in our first trial, our randomization phase was a pretty short term. We were looking at progression over 8 weeks. And what we know now as our SCA trials, our natural history studies, which show us how the disease progresses in patients on no drug, if we're looking at a disease modulating drug, so a drug that's going to attenuate disease progression, we probably need to look at that drug over a longer period of time. So recently there was a publication that looked at all of the studies across the globe. And they did basically a meta-analysis, which is in essence a statistical analysis that combines data from multiple scientific studies in order to derive conclusions about and kind of look for trends in that research area. And this paper showed that overall, if you look at all of these natural history studies of the natural evolution of disease, and you look at it using the SARA score, the scale for the assessment and rating of ataxia, patients with SCA1 progress on average about 1.83 points per year in their SARA, SCA2 about 1.4 points per year, SCA3 roughly the same, and SCA6, a slower progressing disease, about 0.81 points per year. So I put this slide in to illustrate kind of how do these studies help with clinical trials and how do they help guide clinical trials? Well, at the end of our 8-week randomization phase for our first trial in SCA for troriluzole, we had an open label study that lasted initially for 48 weeks or for a year. And we decided at the end of this, well, what can we do with that data? It wasn't a randomized controlled clinical trial, but what can we do? Well, we had, courtesy of all of our colleagues across the country that participated in the US longitudinal cohort, the data from the natural history US trial. And we compared our study with patients on troriluzole to patients on no treatment at all and how they progressed over 1 year based on their total SARA scores. So we ended up matching our patients on SCA genotypes. So we used patients with SCA1, 2, 3, and 6. We matched patients based on age, gender, SARA score at baseline when they entered the study, and initial SARA gate item score. So this is a slide that just kind of illustrates here. This blue arm right here are patients in the natural history study. So these are patients on no drug. This combined group of patients progressed a little over 1 point per year in the SARA, which is what you would expect. This red bar here are patients from the 201 study who were treated with troriluzole over the course of 1 year. And what you see here is that these patients had a very slight improvement or remained at baseline. They did not progress in their disease. So they showed a difference overall of about 1.5 points on the SARA from patients who are untreated. So we were able to use the natural history data to give ourselves compelling evidence to say, okay, now we need to take this drug and look at this over the course of a year in a randomized controlled clinical trial, which is what we currently have running in our 206 study. And I'm just going to go on to just highlight that this also kept us going in the open label phase. Our investigators, our physicians who were running these clinical trials called us and said, look, our patients don't want this trial to end. They want to stay on drug. They feel that they're getting worse when they're off drug. Is there any way you can keep this going? So we added another 96 weeks onto this study in addition. We recently had a full 96-week cut of the data and looked at that, which we're actually going to proceed with a publication for. But just to highlight the basics of what we found was at greater than 2 years. So these patients were on drug for 96 weeks, but remember, a lot of these patients had a gap in dosing where they weren't on drug while we were getting that second phase of the study up and running and getting it approved through the various centers as well as through the FDA. So what we saw was that these patients, even as far out as 3 years, because they had these gaps in dosing, had a less than 0.5 progression on their total SARA. Now, if you remember from that second or third slide I showed, you would expect at least greater than 2 points of progression over 2 years in the SARA score, and we did not see that, which was very encouraging. The other thing that we looked at very closely, because we heard this from our investigators, is what happens when patients come off the drug. And what we heard over and over again was that they were complaining of feeling worse. And that was our impetus for really continuing this open label trial. When we broke down the data and we looked, actually, we have some very nice graphs to show that the SARA scores actually declined in accordance with the duration of the gap. So the longer the patients were off treatment, the more their scores declined. When troriluzole was restarted in that second extension phase, patients actually experienced an improvement in SARA scores, and they returned back to their baseline, which is where they were when they entered that study a couple years earlier. So this is just highlights the design of our current study. So it's the 206 study, which we will look at troriluzole versus placebo in a randomized controlled trial. So half of our patients are randomly assigned to placebo, half of our patients are randomly assigned to study drug, and we're going to look at these patients over the course of 48 weeks. And, you know, again, we're going to talk a lot more during this conversation about outcome measures. In our first trial, we did use the SARA, which was the only validated outcome measure for measuring progression of ataxia symptoms in this patient population. What we realized was that there were a lot of items within this SARA scale that were a little noisy. In other words, while they may be very useful to look at patients over the long term and how they clinically progress, looking at them over a year, it was very difficult to use those items to assess a meaningful clinical change. So in other words, a clinical change that affects a patient's ability to function. So we actually worked very closely with both the FDA and also our physicians that had been involved in the 201 trial, including Dr. Wilmot, who's on the phone, and Jeremy Schmahmann, to come up with a modified version of the SARA scale, which we are calling the modified functional SARA scale. And this, you know, I just added to this slide, since it's within the ataxia space, we also have a myeloperoxidase platform. So we are now investigating therapies for neuroinflammation. So one disease entity we're looking at is multiple system atrophy. So there are two varieties to multiple system atrophy, the Parkinsonian variety and the cerebellar variety. And the cerebellar variety is universally seen in our ataxia clinics. There is a lot of evidence to suggest that the underlying disease mechanism for MSA involves oxidative stress and neuroinflammation. So we're looking at verdiperstat, which is a myeloperoxidase inhibitor. I'm not going to get too much into this trial, just to tell you that this is also a 1-year-long trial, randomized control in a 1-to-1 fashion with patients on verdiperstat and placebo. That study is almost completely enrolled, whereas our SCA study, we are a little over halfway there in our 206 study. So we are still enrolling for our SCA study, SCAs 1, 2, 3, and 6. And with that, I'm going to turn this back over to Dr. Wilmot so we can talk a little bit more about industry and trial development in this space.
Thanks very much. Thanks so much for the perspective from both of your companies. I think that shows how much has been done in this field and how exciting that is. I want to, some questions have come in, a number of them. I'm not necessarily going to address the questions that I see in an individual basis because they're very specific and they can be a little bit hard, but I'm going to try to make a few statements and cover a few things in summary that will give you information that might be very helpful for everybody out there in webinar land. And if you want to write some things down, I'll give you some information that might be helpful, and I'm sure that can be shared later on by NAF if needed. There's been some, and this is all related to the stuff that we just heard about, and we'll turn it back to our other panelists in a minute to comment further. First of all, there's some questions about, is there a trial for this condition or that condition? Is there a trial in this area? Can I get into a trial when I have this? Are there European trials, etc.? And there's two resources that everybody should know about that will give you information about clinical trials. One of them is clinicaltrials.gov, G-O-V. Clinicaltrials.gov is a governmental registry of all treatment trials. If you are doing an intervention and a clinical on humans, it must be listed there. Other clinical studies that are simply observational, where we, for instance, with an ataxia, follow patients along over time. Many of those are also, but don't do an intervention, no treatment necessarily, but just follow patients over time, might also be listed on the clinicaltrials.gov, but it's not as strict a requirement as with the interventional trials. And then for Europe, there's clinicaltrialsregister.eu. And I don't know as much about how that works, but it seems to be very similar as clinicaltrials.gov. So I just encourage anybody who wants to know a little bit more, you can search, you put in ataxia or Friedreich's or SCA or DERPLA or whatever disease you have, and you can find out what studies, particularly intervention trials, but maybe other studies as well, are available for you to potentially participate in. Can everybody who has that condition participate in a trial? And the answer is no. There's a couple of issues that I want to talk about for a second. One would be inclusions and exclusions. That when we talked about designing a clinical trial, when Melissa was talking about that, the idea is we want to try to do a trial in a way that we can have good chance of finding an effective treatment if that treatment is effective. We don't want to be limited by other outside factors, such as including everybody, including some people who cannot improve because their diseases, manifestations aren't as measurable, for instance. So we have to be able to measure ataxia. We have to have patients that have measures of the ataxia in a certain range where we're likely to be able to find differences over the time of the trial. If we can't find those differences, even with an effective treatment, we might not be able to see the effectiveness in the trial. So these are all considerations that the people from industry have to think of very closely, because they want to, be able to develop something that is helpful for the patients out there. So the inclusions and exclusions are very important, and those are usually listed on those registry pages that we talk about. I think we've heard a few things that are challenges in developing treatments for ataxia. And we saw the wonderful timeline that Mike presented about coming up with compounds. That's one challenge. You know, just getting the right compound to test. Having a good measure of ataxia is another challenge. Being able to recruit patients. You know, do we have enough patients out there to be able to recruit? Those are all things that you guys, in running a trial from industry, have to deal with. But there are also frustrations for us as investigators, and there are frustrations, I'm sure, for patients as well. So let me ask you guys as panelists, what do you think, or what has in your work so far in ataxia trials, what has been the most challenging thing that you've had to overcome? Mike, you want to answer first?
Yeah, I think first, as we discovered 1883, I think it was the translation of the models, right? So how do we know we have a drug that can treat ataxia? Well, we use rodent models, right? We make genetically modified models, and then we hope that the positive response that we see in the rodents will translate into a positive effect in patients. And it's very difficult to measure ataxia in these models. And to Melissa's point, there is no approved treatments in spinal cerebellar ataxia specifically, and even broadly in ataxia, there's not that many, right? And so we don't know if the models that we're actually using to pick the drugs are telling us the right things, right? So we have to take a bit of a leap, right, to say, okay, we can fix the disease in this model. All right, let's go to the clinic and see if we can actually improve disability in patients with disease. So I think the translation of models is really big. And then I think the other point is endpoints, right? I think SARA has been around a long time. People worked really hard on it. It can clearly measure disability over time. Our challenge, and I think most companies that get into the space for the first time, is the ability to see a response in a short duration. I think Melissa laid out some really interesting data over a couple of years. That trial is hard to get to if you don't see something, right, in the early stages, right?
And so you're leaving out an important. Why can't you just do a 5 or 10 year trial and see if it works?
Right.
There's several reasons. You know, with a new molecule like 1883, so 1883 never existed anywhere until we made it. So, and we know the safety of that molecule. For if I give it to animals for 12 weeks, things look pretty good, right? I need to continue to establish that safety profile over time, right? A drug like troglizol, right, has the advantage of leveraging years of experience with riluzole, right? So that's been in patients, lots of patients, right, for many years. So the safety of that drug is a little more well known. Our drug, we're building that safety all along. And the first thing that we want to ensure is that we don't do any harm, right? So we need to make sure our drugs are safe. We know today our drug is safe for 12 weeks, and we're going to have to continue to build on that safety package. So to get to a 1 year, 2 year, 5 year study, we need to go through 12 week, 6 month, 9 month. We need to go slowly to ensure safety. And then all along, we really need to see some hints of efficacy. And that's why we call our first trial a proof of concept, right? We're not looking to cure ataxia in the first trial, but what we are looking for is encouraging signs to say, your hypothesis was right. If you can improve Purkinje neuron regularity, you're going to improve disability. Now keep going, right? And so that's so important for us, which is why, you know, I think SARA is a great endpoint over time, and it's really shown to be useful in longitudinal studies, but it's relatively insensitive, I think, to smaller changes, which is why we're like focused on sway and turn. Like if we can see improvements in those, then we're emboldened and encouraged to run those longer term studies. So in addition to the clinical piece, we need to do a whole bunch more safety studies in animals before we can do those longer term 1+ year studies.
Right. Let me just point out one thing that might not be clear to some of the listeners, but there are 2 ways of treating ataxia. One is symptomatically, if you make the movements better. The other is protecting the brain from ongoing damage of the disease. We call that neuroprotection. And neuroprotection, you might not, ultimately will make somebody who's on a good neuroprotective agent better over time, but might not have a symptomatic effect on the actual ataxia that you're having, like at the moment, if it doesn't have a symptomatic effect. And it takes a lot longer to look for that neuroprotective effect, and a shorter symptomatic medicines are more likely to show up when you're doing a shorter trial. So, we have to deal with what effect are we expecting. It's a little bit complicated because if you are symptomatically benefiting patients, maybe that's also long-term being protective, and maybe that's what you think you're seeing, Melissa, with some of the shorter term and the longer term results that you've seen.
Yeah, that's right. And you know, in this disease space too, right, as we've been talking about with endpoints, it's much different from, say, Alzheimer's. It's a disease that's been well studied over the long haul. The natural history studies on the evolution of Alzheimer's disease are much better characterized, right? You measure, there's biomarkers, right? You can measure amyloid and tau. There are quantitative markers of improvement in the disease process. So when we came into this space in SCA, none of this existed. So in essence, we're paving the way for that, right? Alzheimer's didn't just have that when they started, right? This was hard work by researchers out there in the academic field and researchers out there in the clinical field to pave this way for smooth clinical trials. The initial clinical trials really had to go in at risk and figure this out as they went, which is where, you know, it's interesting because, you know, some people will say, oh, a clinical trial didn't hit its endpoint, oh, that's a waste. No clinical trial is ever a waste, because even if you don't hit your endpoint, you learn so much about disease progression. And if you include the right exploratory endpoints in your study that you're looking at, you also learn a lot about other scales that you could potentially use in future clinical trials.
Right. So we talked about those issues. Melissa, did you want to say anything else about what you foresee as one of the biggest challenge to doing clinical trials in this ataxia world?
I mean, I think the two challenges in any rare disease space, especially in any neurodegenerative disease, right? We've already touched on the endpoints up, down, and sideways, but also there are rare diseases that can be difficult to recruit for, right? So when you're recruiting 230 patients across the country for your trial, which, you know, when we design clinical trials, the reason we have a certain number of patients we need to participate in this trial is because we need to sort of power that study for statistical significance. We need to have enough patients in the study that we can be sure that when we reach our endpoint, we had enough patients to prove that that was actually statistically significant. And that's a challenge as well, especially with the SCAs, because, you know, our 201 study, it was a lot easier, as you know, because we had, you know, the 18 most prominent neurologists in ataxia across the country, and their patients entered the study, right? So now the 201 patients cannot enter into the 206 study. So we're going for a completely different pool of patients. And a lot of patients with SCA, even now, especially in rural communities, which, you know, brings us to the fact that there's not enough awareness about the disease. They haven't even had genetic testing. So they don't even know if they can participate in the study because they've never been tested for one of the autosomal dominant ataxias. Right. And that's been a challenge as well. And I think that, you know, again, it all ties into how young this field is. But as we've seen in the last 4 years, it's kind of exploded.
Yeah, definitely. So what about, what are there some concrete ways that patients or advocacy groups or physicians or you know, anybody can do something to help your efforts in trying to find an effective treatment for ataxia? What do the rest of us need to be doing that, you know, I mean, hopefully we're doing some of that now, but there's probably more that we can be doing. Any other ideas about what more that can be done?
You know, from our perspective, you know, getting back to your point about eligibility criteria and understanding the population, right? So what patients are out there, right? And so I think the registry initiatives and activities that the NAF is involved with, I think are really important, because as we think of ideas and not even just, you know, Biohaven and Cadent, but the other players in the space, you know, they really need to understand what the population is, right? Like how many patients with the different genetic versions of SCA, what kind of disability are we talking about, right? And I think the longitudinal studies are really helpful in defining that progression. I think that Melissa showed in how Biohaven's using it to show that their drug is slowing down progression. So I think participation in those registries as a patient is really important to us, because we keep coming back to, you know, all the KOLs in the space, asking them specific questions about their population, and then they'll often say, well, that's a good question, we should add that to the registry, right? And so I think participation in the registry is really, really helpful as we think about trial designs. And then maybe it's a thank you and encourage folks to participate in trials, these non-interventional trials that we've done at MGH, and I know there's more queuing up from some of our other colleagues in other companies to try to understand disease progression. And I know there's lots of work now starting to work on biomarkers to try to, as Melissa mentioned, some of the markers in Alzheimer's, to try to establish those same kind of benchmarks and biomarkers in ataxias, because they're super helpful in looking at disease and trying to figure out if your drug's doing anything. If you can see those markers moving, you're encouraged to keep going. And I think participating in those kind of trials, although they're non-interventional, right, they're really important to establishing a baseline to help us understand what this disease is all about.
Okay, good point.
Yeah, and I'll just say on that too, when you design these clinical trials, and Mike can speak to this too, it's very tricky because you have to align the investigators that are participating in the trials, our doctors who are actually running the trials out there in the field, but we also need to engage the regulators, which in this country is the FDA. So everybody kind of needs to get on the same page. And especially in clinical trials, the FDA kind of guides us towards what they feel are good outcome measures for clinical trials. And I actually think that, especially in recent years in the ataxia space, we see people delving into every single one of these. So the natural history studies, right? The longitudinal studies, looking at patients off drug, how they progress. Patients participating in those studies is incredibly important. And PIs, investigators, physicians conducting those studies and being involved in them is really important. The quantitative assessment studies, Mike's, Dr. Chris Gomez, who's one of the leading ataxia specialists in Chicago, has been running a biosensor study for quite some time. There are a number of biomarker studies. And then one big trend right now with regulators is the patient voice. So patient-focused drug development and patient-reported outcome measures. So at Mass General, Dr. Schmamina, his ataxia unit, he is developing the first patient-reported outcome measure for ataxia that actually we've been lucky to kind of work with him to help him validate. And that has been hugely successful. And I think that's going to be now pretty close to being ready to actually put into a clinical trial as an outcome measure. Patient-focused drug development groups, NAF is now going to be hosting some of the first externally led patient-focused drug development groups in ataxia. That's going to be huge. And the summary of those patient-focused drug development groups, something called the patient voice, really informs us about what clinically is important to patients. So when we're measuring outcomes at the end of a clinical trial, we need to know those changes that we're seeing in a patient's clinical life, that's important to the patient. That changes their ability to function in life, and that's very meaningful to them. So I think, and awareness in general, which I think, our colleagues at NAF have been phenomenal in, and there are even some, young people across the country trying to establish social media sites. We try to do that through Biohaven. I know Cadent's trying to do that too. It's really a collaborative effort, really, of getting patients, industry, regulators, and advocacy groups all together working towards the same common goal.
Right. There's a few questions that are on here that I feel like we should address, or that, honestly, I can just address them pretty quickly, but I want to do that. One is about how do you volunteer for a SCA study? It depends on what study you're talking about. There's the natural history study where we just see how the disease evolves over time. And there are sites that are doing that. Information for that is on the NAF website. And NAF is always a central hub for all of these studies because they know what's going on. They know the companies that are involved. And so you can utilize their expertise. It's not a huge group, and don't overwhelm them too much, but I think that would be a good kind of central place to find out more information. You can volunteer, for instance, for a Biohaven or a Cadent study, but if you don't meet the criteria for it, you're not going to be necessarily accepted. So it isn't like if you want to participate, you absolutely can. You have to meet certain criteria. And to find out about those studies, again, you can go to clinicaltrials.gov, and when you're on there, it will list all of the sites that are active in that trial, and a coordinator, a contact person, and you just call somebody up and say, hey, would I qualify for your trial? There's also, and there's a few questions about drug treatments if you don't have a known ataxia or you're undiagnosed, and then some even if it's acquired. And if you know that it's acquired and it's due to a certain cause, you try to deal with that cause. Like if you have MS causing ataxia, you deal with your MS, for instance. But that's a little different situation. For undiagnosed ataxias, the participation in trials might be limited because, again, the trial is to try to find a good result if one exists, and that is reliant on having a really defined patient population as people in the study. But you can potentially, this is the two things. Number one, we would love to test like everybody with undiagnosed ataxias and find out who is SCA positive, and then be able to direct them towards Biohaven or towards Cadent or towards somebody else, when they're ready to do that, and that type of thing. And so there are some efforts that are ongoing about how to try to do that, because testing, unfortunately, as Melissa alluded to, just doesn't always get done for insurance reasons and for other reasons. And if we had a way of trying to make that, tenfold more common than is currently done, boy, that would be great. We would definitely find more people. And if you had an undiagnosed ataxia now, you might find out actually what the diagnosis is, and then it could give you direction. And the other thing is that the other point I want to make was the medicines that they are developing, let's say that it's an SCA2 patient and Cadent is doing something on it and has a positive result. Let's say that Biohaven has a positive result in it. That's great. You'd have 2 drugs, and let's say that the results are good enough, they go before FDA, they get approved. Now there's 2 things, but what if you don't have SCA2? If you don't have one of the ataxias that's on that list of a medicine that is approved for ataxia, can you take that medicine? And that opens up a whole can of worms that we're not going to get into right now. But all I would say is that in research, we always learn so much about one disease, even when we're studying another disease, that we can learn something. Like, for instance, there was a question about antisense oligonucleotides, ASOs, which is one approach to some of the trinucleotide repeat diseases like the SCAs, some of the SCAs. It's been tried in Huntington's disease and in other conditions. And there are definitely companies that have programs developing that for SCA, though there's no clinical trials going on right now in that. But what we have learned from the ASO trials in Huntington's disease, which is not ataxia, is applicable to us, which is great. So maybe there's an ability to use a treatment that was developed for something else in your particular disease, maybe not. But in any case, there's always a wonderful spread of knowledge that helps everybody. And that's a very good thing we all recognize and bank on. Choosing site for clinical trials like weakness. So do companies offer free genetic testing? Great question. Not really. There are ways of, there are some ways of getting testing done for very little money, actually. And there's some testing that's done potentially within a clinical trial. If you have a definite SCA, meaning an autosomal dominant inherited ataxia, it might be possible to get you tested within the confines of seeing if you're eligible for a trial. I don't know if either of you two want to talk about that, but just, you know, free testing available on the street corner from your local doctor, nah, doesn't really exist. Though I would love it if that type of thing were some way possible, and maybe there'd be some way of doing it through some collaborative effort, but right now that is not completely implemented.
Yeah, well, I can speak to the Biohaven trial, Chip, as you know, because you have patients enrolled in it, right? So, you know, if a patient wants to be screened for the trial and they have a pattern of an autosomal dominant ataxia, Biohaven pays for that panel of testing. So that is a way to get, you know, testing done.
Yeah.
But I think there needs to be broader access to testing across the country, and that's going to come with, you know, increased funding for the disease entity entirely, right, and more awareness. So I think even in the last 3 years, I know 3 years ago I was at an ataxia walk that Jeremy was hosting, and there were, you know, 4 patients who were local, you know, they were in New England, who came to him teary and said, and they actually did have, one had an autosomal dominant pattern, but all 3 patients had seen clinicians in this area, neurologists, who had said, you know, it really doesn't matter what your genetic test turns out to be. There are no treatments, so it really, really doesn't make a difference. And if we're hearing that in places like Connecticut and Massachusetts and New York, you know, what are people hearing in rural communities out in, say, the Midwest, right? And I think we know for a fact that they often don't even have testing available to them, and they hear over and over again, it doesn't matter. But what we know is it makes an enormous difference, because whether there's a treatment or not a treatment, to have a diagnosis means an awful lot to a patient. And now that there are clinical trials in the ataxia space, it makes a big difference as to what you're eligible for and what opportunities you have. And so that needs to change, I think, simply through awareness. As that changes, I think testing will become more readily available.
Yeah. I will just say, as a ataxia clinician, you know, outside of the trials and all of that, the issue of genetic testing is just, it's a huge one, and it's hard to deal with, certainly from the families, from the clinician standpoint, and it's an important one. I just wish that basically anybody with ataxia, because there's a lot of genetic ataxia out there that might not know that it's genetic. You know, when you have a very strong family history, it's very obvious. But if your father died young and you're not very close to the rest of the family, maybe it's very possible that there's an obvious family history if you knew it. And then there's also, you know, possibilities for other reasons to have genetic causes like new mutations or anticipation, et cetera. So wish we could test everybody, and it just doesn't work out that way right now. But we're all kind of working towards wanting to do that. We are at the top of the hour, but I know that we can stay a little bit longer. I wanted to, first of all, just cruise by the questions and make sure that we've answered most of them. There's a question about acetyl-D-leucine, its availability. I don't really know. I know there was a trial. I don't really know the details on why it was thought to maybe work. There's some model systems and stuff, and there's been things with some amino acid derivatives like that, the serine and serine and stuff. So I'm not going to really speak more on that. Don't know a whole bunch more about that. Let's see. I would like, if my understanding is accurate, I have unknown type, any current research involving medication. Yeah, so I am unaware, again, might need to go to clinicaltrials.gov, but there, the question was, if I have an unknown type of ataxia, because of these inclusions and exclusions, do I qualify for any clinical study? And we kind of dealt with that with the testing. If you were tested and turned out to have an inclusion or type of ataxia, in other words, an STA1, 2, 3, ARCO1, 1, 2, 3, 6, 7, 10, 8, then yeah, maybe there's a trial available for you now. The other thing is that if you didn't have that, let's say that those were all negative, there are sometimes small trials that are done more institutionally, not huge, big collaborative efforts that are very expensive, a lot of which are driven, by pharmaceutical industry for that reason. But also, there might be some smaller trials that are available at a local institution. We'll see, for instance, University of Florida did some stuff with 4-aminopyridine. There was the small trial out of USF on varenicline in the past. There are things along, and with a little bit of other people recruiting for too. But so there are those types of trials that sometimes will include people who don't have an undefined type. I don't really know of anyone going on right now, but honestly, sometimes those types of things will pop up without me really hearing about it. And NAF might catch wind of it. You can always check, again, clinicaltrials.gov. And I do that once a month at least. I just go on there and search ataxia and kind of see, am I missing anything that I didn't know about? And when you do that, you might end up finding something. But I think, again, the biggest hope is that we can do more testing and find more people who have types of ataxias that do qualify them for the trials right now. Or that we can get positive results and then we can test them in unknown ataxias. Anything for leg weakness, that's too vague a question. Or I don't know the context with that. So there's no medicine that I know of that actually, clearly simply without side effects and stuff increases strength. Mostly that's a physical therapy kind of thing. Okay, I don't really see any other questions that we need to address. Mike or Melissa, because we are, a little bit over time now, but did you have any last things that you wanted to say to the folks out there?
I mean, I can start, and maybe I'll start again with thank you for helping us as Cadent kind of understand this field over the past 6-7 years, right? And we are kind of in it with you in the sense that we want to bring a drug that helps improve your life, right, and helps reduce disability from this disease. And helping us understand what that means is really important. And getting back to Melissa's point about the regulatory endpoints and the FDA, I think at the end of the day, we need to convince the FDA that the improvements that we have in patients with ataxia are meaningful, and that the drug will make a difference, right? And I think the PROM is really important. I think the work that's being done there, those take a long time to make happen. And we're really impressed with the work that's been done already and the patients that participated in generating that PROM. It's going to be really important as we get these trials off the ground. So a big thank you, and I just encourage folks to continue to participate because there is a lot of activity, even beyond the activity at Biohaven and Cadent, of companies getting into the space with new ideas of how to treat these diseases.
Yeah, and I'd like to echo that, especially with a thank you to our patients out there who are working in advocacy groups and also participating in natural history studies, non-invasive studies out there to advance research on ataxia and these clinical trials. Every little bit plays a role, and I think, we can see how a field can explode, and sometimes it happens because a celebrity gets a disease. I mean, in all honesty, right? When a few celebrities ended up with Parkinson's, that field exploded. But I would like to leave this on an optimistic note that, in the time that Biohaven alone has been in this space, the advances have been enormous. The advances we've made with outcome measures, the advances we've made with drug development involvement, right? Involvement of pharma companies. The first NAF meeting that I ever went to through Biohaven, we were the only pharma company there. And the last meeting I went to, there were numerous pharma companies there and numerous people interested. So every little bit propels you. You get one clinical trial under your belt and other companies become interested in this space. and you get a lot more awareness out there so that patients know what centers to go to get genetic testing, what centers to go to get a definitive diagnosis. And every little bit of that helps advance the field. But, you know, in what I alone have seen in the last 3 years, and, you know, Chip, you can probably comment a lot more on that because you've been in this field for a really, really long time. The last couple of years have been impressive, and I think the future is bright for this field.
Yeah, no, I think what I've seen over time has been just amazing, and it's the big shift that's been over the past few years is that shift to industry involvement, which is just wonderful. You know, when I started off in Ataxia, we didn't even have the SARA or good measures. There was certainly no drug that was ever, you know, even thought about as possibly being helpful. And I tended to think of the pharmaceutical industry as those folks over there, you know, who, you know, they're going to charge a lot for their drug, and they don't, you know, they're not necessarily the good guys. And boy, have I changed my view on that, because I've seen the benefit of having the pharmaceutical industry developing trials. We as researchers would never have the resources and the knowledge and the ability to deal with regulatory problems and everything like that to develop drugs ourselves. That's such a hard thing. We need the pharmaceutical industry to be involved, and the fact that it is with such excitement and collegiality and true good motives to try to bring treatments forward is just really, really exciting to me. So thanks everybody for tuning in. Lori, you have some closing comments?
I do. Thank you so much, Chip, for your expertise and experience as one of the leading ataxia clinicians and researchers, and bringing that to this conversation. And to Melissa and Mike, thank you so much for the work that your companies are doing in this space. Where really, it was great to learn about all of the years of work that is behind your projects, and we're really excited about the therapies that you're working on. So thank you. And I wanted to also, for our attendees out there, let you know that we're going to continue to provide education and conversations and opportunities for you to continue to learn about different things going on in drug development and ways that you can participate and be a part of this, because we certainly need all of your participation as research collaborators in this process. And with that said, I did put a couple of links that Dr. Wilmot had referenced from NAS website and clinicaltrials.org, and also wanted to let you know that we are having a patient-focused drug development meeting with the FDA virtually on September 25th, and you're all invited to tune into that exciting event. And with that, I just wanted to again thank all of our attendees for joining, thanking our great panel today, and I hope you all take care and stay safe. Thanks, everyone.
Thank you, Lori.
Thank you.
Thank you, guys.